just when you thought it couldn't get more complex, let's look at complex inheritance patterns. Okay, so Mendel said that there's a law of dominance, which means that some alleles are dominant and others are recessive. And then the law of segregation stated that organisms, they inherit 2 copies of each gene, one from each parent. And only one passes on 1 copy of each of that gene to their gametes, to their sex cells that will become offspring. And the law of independent assortment says that genes for different traits assort independently during the formation of gametes, causing an even more randomization of eligible offspring. There's a theory of chromosome theory of inheritance. So genes are located on a chromosome at one location, and the behavior of chromosomes during meiosis account for inheritance patterns. So there's more to this than just those 3 rules. We have to have exceptions or some differences depending upon which chromosome we're talking about. Because remember, they're all different sizes, so they can behave differently. And of course, you have those sex chromosomes where females have X and X and males have X and Y, so they are different. There's exceptions to Mendel's law. So some of those alleles, they're neither dominant nor recessive, and many traits are controlled by multiple different alleles. In other words, multiple genes control traits. You would expect that, right? Life is complicated, and so is our DNA. So if you think about your own traits, which one do you already know that don't fit within Mendelian genetics? Based on the picture of kitties that we see here, you can tell that everybody's got blue eyes so far, but some of them are patterned differently. Not only that, but there's some different coloring too. We have orange cat with white stripes, white cat with black stripes, orange cat with black stripes. So just by looking at the photo, you can tell that there's more than just one gene that's controlling hair color in cats. Incomplete dominance is a heterozygous phenotype is somewhere between the 2 homozygous phenotypes. So neither allele is completely dominant or recessive. A perfect example are the flowers that we see in these snapdragon flowers. So we have the red, we have the white, and we have the pink. Some might say that the pink one is a hybrid of the other 2, when really it's just the color red is incompletely dominant over the white. So if you remember our crosses, if I have a red plant and I cross it with a white plant, then all of my offspring will be pink. They're all going to be heterozygous compared to their homozygous dominant red flower and homozygous recessive white flower, creating this incomplete dominance of color, which yields pink flowers. Let's do an example with incomplete dominance. So in humans, curly hair is homozygous dominant. It's incompletely dominant to straight hair, which is dominant H prime, dominant H prime. The heterozygous individual has wavy hair, H and H prime. So let's cross 2 people with wavy hair. When neither allele is completely dominant over the other, we technically have 2 dominant alleles. We call one of them primed because it will eventually be masked or mixed. Okay, so we are going to cross these 2 wavy-haired people, and in our Punnett square, we are going to just fill this in. And what we get is A ratio of 1 to 2 to 1, or 25 to 50 to 25, and curly hair, which is HH, so they are 25% likely to be curly, 50% of the gametes produced are likely, or I'm sorry, of the offspring produced are likely to have wavy hair, and 25% are likely to have straight hair. So our genotype ratio will be a 25 to 50 to 25, and we simplify that down to a 1 to 2 to 1, curly, wavy, straight. Let's do another example. It says Fitz loved growing flowers for his friend Olivia. Her favorite flower, which are roses, are found in red, homozygous dominant, blue, R prime, R prime, and purple, heterozygous, R and R primed. So what would happen if Fitz crossed blue roses and purple roses? So there's our blue rose, and then the purple rose is R and R primed. And when we fill in our Punnett square, we have a 50-50 ratio. So this is a genotype that is 50% to 50%, or a 1 to 1, R primed to R prime, R prime. And then a phenotype ratio is going to be, right, 50 to 50. That's a 1 to 1, or half of the offspring will likely be purple, and the other half are going to likely be blue. Notice how we didn't make any red offspring.